Right, so pathogenesis of infectious diseases, as I mentioned again, all of this information you would have already been exposed to, so I'm going to go through pretty quickly, talking about some definitions. This part of the lecture has just a lot of definitions introducing us or reintroducing us to what infectious disease is and what types of infectious diseases people can have. So whenever we hear the term pathogenicity, it refers to the ability of a microorganism to cause diseases. And pathogenesis refers to the steps or mechanisms of how a disease develops over time. So as we know, not all microbes are pathogenic organisms. There are only a very small number of microbes comparative to all that are present that can cause infections. A lot of them are helpful and so on. We spoke about those things earlier in the semester. So when we hear the term infectious disease, it is a disease caused by a microbe, and all of the microbes that cause infectious diseases are collectively referred to as pathogens. So we have microbes for the common term that is used for microorganism. Those that can cause a disease are called pathogens. The term infection is something commonly used to talk about what types of things we suffer as an effect of that infectious disease. So for example, when somebody says they have an ear infection, it basically means that they have an infectious disease in their ear. Okay, so it's used as a synonym. Microbiologists usually reserve the word infection to mean colonization by a pathogen. The pathogen may or may not go on to cause a disease. A person can be infected with a pathogen but may not have an infectious disease. So infection does not always occur for several different reasons. Sometimes you may have the microbe landing on a site that it cannot multiply. So if it is a microbe that is causing a disease on the respiratory tract, if it lands somewhere in the digestive tract, it's not going to be able to cause a disease. If it's something that causes an infection of the eye and it lands on near the surface of your skin, it's not going to be able to cause a disease, right? Many pathogens must attach to specific receptor sites, and because if they are not, they cannot multiply and cause damage, so infection will not occur. There may be antibacterial factors present at the site where the pathogen lands that can prevent the organism from causing infection. Sometimes there may be indigenous microflora, which are microflora that live on our bodies, that may fight off that organism where it lands so that it does not cause infection. Because remember, obviously, that microbe that lives there regularly does not want this other invading microorganism to take over its habitat. Sometimes the indigenous microflora may even produce toxins or antibacterial factors to destroy the pathogen. And that's why they always say you must be careful what types of substances you apply to your skin, creams, and so on, because you do not want them to affect how those indigenous microflora work, because sometimes they're obviously very helpful to us. They can stop other organisms from causing infection. The individual's nutritional and overall health status can also influence whether pathogens can cause disease. The person may be immune to that particular pathogen, so sometimes we would have gained immunity by previous contact with that pathogen so that they cannot once more cause infection. Or you can have phagocytes that are present in your blood internally that will destroy the pathogen. So there are 4 phases that occurs during an infectious disease. First, you must be exposed to the pathogen. The pathogen must be able to be incubated at that specific site, and then they go through a prodromal period, a period of illness that would lead to either death, disability, or convalescence, right? Localized versus systemic infections. As the term suggests, a localized infection is an infection that is present at some specific region. So once an infectious process is initiated, the disease may remain localized. So for example, a pimple or a boil that is located in one specific region of the body, so that they are called localized infections. They don't affect other parts of the body. You can also have systemic infections. This is where it spreads throughout the body, and most of the times these types of infections are transported by the blood so that they enter all of your different systems and can cause multiple infections at multiple sites, very harmful. Then you can have diseases classified as either acute, subacute, or chronic. Acute diseases are those that have a rapid onset, followed by a rapid recovery. Example, measles, mumps, or influenza. A chronic disease, very slow onset, and it can last a very long period of time. So for example, you can come into contact with this disease at a very early stage in your life, and they don't start showing signs and symptoms until 10 years after, maybe, right? Examples: tuberculosis, leprosy, syphilis. Or you can have subacute diseases. This is in between those two categories. It comes on a little more suddenly than a chronic disease, but less than that of an acute. Example: bacterial endocarditis. So, signs versus symptoms, something that's very commonly confused, right? But they mean two different things. A symptom of a disease is defined as evidence of the disease experienced by a patient. So something that is subjective. So for example, aches or pains, ringing in the ears, vision, blood, and so on. So different people have different symptoms. Somebody who is older would generally have more severe symptoms than somebody who's younger and more capable than fighting off an infection. So that although they may have the same disease, the symptoms of the older person may be more dominant than that of someone who is healthy in a healthier, younger state, right? There are some diseases that may be symptomatic, some may be asymptomatic and very difficult to identify. A symptomatic disease is where you experience symptoms, asymptomatic you do not. A sign is different from a symptom. A sign is something that can be measured. It's an objective evidence of disease. So for example, you measure someone's blood pressure, you listen to their abnormal heart sounds, you look at lab results to determine whether or not something is causing them to actually have an infectious disease present, right? Abnormal pulse rate, abnormal lab results, and so on. So a symptom is a feeling or a subjective thing, whereas a sign is something that's objective. It's a measured change or evidence of a disease being present. So don't confuse the two. You all should be able to define both of them accordingly, right? Latent infections, another definition. Latent infections are those that can go from being symptomatic to asymptomatic and then again symptomatic. So sometimes in these infections, people get symptoms, they go to the doctor, they treat it. They go into a stage where they do not show any more signs and symptoms. They don't feel anyhow, so they think that, okay, they don't have the disease anymore. However, it may be a situation where the disease has gone into a period of latency, and later on, at some later stage in their life, they go back to being symptomatic, not realizing that the disease was present internally all along and it was multiplying and it could cause a much more severe secondary infection, right? Example of this is syphilis. Primary syphilis, where you start getting signs and symptoms, can move into secondary syphilis. Then it goes into a period of latency. You have no symptoms. It may last for weeks to years. Sometimes in some patients, it continues throughout their life. And at a very later stage, they start to experience tertiary syphilis, where it comes out from the latency period and it shows up as different types of infections in the cardiovascular system and so on. Obviously, after being incubated for such a long period of time, it would be extremely harmful. It can sometimes lead to death. So, what are the steps in which infectious diseases occur? First of all, in order for an organism to be pathogenic and to cause, or steps in pathogenesis to create a disease, it must be able to enter into the body. It must be able to attach to a suitable site and multiply. It should be able to invade or spread in order to cause that infection. And it must be able to evade host defenses. So even if you have phagocytes trying to attack it to fight it off, it should be able to evade those types of mechanisms so that it survives and it causes damage to the host tissue. That is the entire sequence by which pathogenesis would take place. Another definition of a term, virulence. When we would have mentioned, I would have said that term during the semester, a virulent organism. The term virulent is sometimes used as a synonym for pathogenic. There are organisms that may be virulent or those that may be avirulent in different strains. So we have thousands of strains of the influenza disease. Some of them are pathogenic or virulent, meaning they are able to cause a disease, while others are non-pathogenic or avirulent. Right? For example, you have strains of diphtheria, Corynebacterium diphtheriae, which are toxigenic, or you can have those that are non-toxigenic. So they may or may not cause diseases based on the strain. Sometimes the term virulence is also used to express or measure the degree of pathogenicity. So you can compare organisms by saying how infectious they are. All right. Example, it takes 10 Shigella cells to cause shigellosis. So that disease takes just 10 of the organisms to cause a disease, compared to Salmonella, where it takes between 100 and 1,000 Salmonella cells to cause salmonellosis. So therefore, Shigella in this case is going to be much more virulent or much more pathogenic because it can cause disease with a much lower number of cells compared to how Salmonella works, right? Example, you have different strains of Streptococcus pyogenes that may be more virulent than others. Some strains of Staphylococcus aureus can produce toxic shock syndrome, so they are considered to be way more pathogenic than others that just simply cause little infections. So how do organisms have these different levels of virulence? There may be different virulence factors that attribute to them gaining specific infectious capability and allowing them to become stronger types of pathogens, right? In this case, these virulence factors may allow them to attach more efficiently. They may escape destruction and cause disease more efficiently, right? And there are different types of examples of these virulence factors. These are usually phenotypic characteristics that are gained through genetics. So you can have organisms that gain genes from other types of organisms through conjugation. They bind to bacteria, and the bacteria are able to pass genes from each other to allow them to gain these virulence factors. Examples, they can gain genes that allow them to produce adhesins. These are special molecules on the surface of pathogens that allow them to specifically bind to host cells, so it allows them to increase efficiency of infection. They may gain pili or bacterial fimbriae. These are also considered to be virulence factors because these pili allow them to attach more significantly or more securely to the surface of host tissues. Therefore, they have a much more likely chance of causing infection. So they gave an example here of adhesins, showing you how the adhesins allow them to specifically bind to the host cell because the host cell has specific receptors. They showed you in this example here, they're showing you the pili. There are other virulence factors, example flagella, that may make them swim faster or move faster through environments. They can have capsules that allow them to be camouflaged so that your white blood cells your phagocytes do not attack them, right? They can have different types of enzymes. So they listed a range of different enzymes. They're coagulase, collagenase, kinase, all of those enzymes that may also act as virulence factors because it may allow them to fight off antibacterial substances or some type of control mechanisms. It may allow them to create enzymes that will allow them to easily attached to host cells and so on, right? Then you have obligate intracellular pathogens. As the term suggests, obligate intracellular pathogens. These are pathogens that must live within host cells in order to survive and multiply. So obligate intracellular. Intracellular means that they must live inside of our cells. That's the only way they can survive, hence the term obligate. You can have intraleukocytic pathogens. These pathogens these are organisms that live inside of white blood cells. Leukocytes are white blood cells. So pathogens that are intraleukocytic are pathogens that live inside of white blood cells. You can also have intraerythrocytic pathogens. These are pathogens that live inside of red blood cells. Some examples of that is Plasmodium, which causes malaria, Babesia, which causes babiosis. You can also have facultative intracellular pathogens. These basically, facultative always suggest that they can live in either or environment. So they can live intracellularly or they can live extracellularly. And there are several things that these microorganisms that may gain to allow them to survive intracellularly. So for example, they may possess cell walls that resist digestion. They may be able to fuse lysosomes with phagosomes, prevent that fusion so that phagocytes cannot attack them. They may be able to produce phospholipases to destroy phagosome membranes, to prevent, again, phagocytes from destroying them, or other unknown mechanisms that allow them to survive the host defense mechanisms. Capsules and flagella, as I mentioned previously in the diagram, they are also virulence factors. Flagella allow them to move very quickly in aqueous environments so that they can invade and infect hosts very efficiently. Capsules usually allow them to camouflage themselves so that phagocytes cannot recognize them. You can also have exoenzymes, as I mentioned, that are released. The major mechanisms by which pathogens cause disease are the exoenzymes or toxins that they produce. So these enzymes can give them the ability to create specific toxins that are responsible for infections. Toxins are poisonous substances that are released by different types of pathogens. There are two major types, endo and exotoxins. Endotoxins are part of the cell wall structures. They are released externally on the outside of those pathogens. They can cause things like fever and shock. Or you have exotoxins. These are secreted, poisonous proteins secreted by pathogens. For example, neurotoxins, enterotoxins, exfoliative toxins, and so on. So not only can the pathogens have virulence factors that allow them increased rate of survival, they can also escape immune responses by several different things. They can have antigenic variation, where they can change their surface antigens to resemble a host cell, so that when your phagocytes are basically circulating your blood, they do not recognize these pathogens as harmful, right? They can camouflage themselves, or they can mimic molecules. Some organisms can conceal their foreign nature by coating themselves with host proteins like a camouflage mechanism. Again, they mimic what host cells look like, or they mimic what microbes look like that we are accustomed to in our bodies, so that our pathogens, our phagocytes and our immune system do not recognize them as foreign, therefore they do not fight them off. Or you can have disruption of antibodies. There are some pathogens that produce specific antibodies or enzymes that help them to escape from antibodies present in the host cell. Okay, so that was a bit of introduction into your topic today, which is talking about your immune system and pathogenesis. And that will lead us into talking about non-specific host defense mechanisms. And again, all of this would be a revision, right? Some of the information in this part of the lecture would be a little new. You would have been exposed to it before, but not in too much detail, right? So, host defense mechanisms are defined as the ways in which your body protects itself from pathogens. There are three lines of defense that we have against pathogens. The first two lines are non-specific, meaning they do not target specific organisms. They just kill off or block anything that is foreign. The third line of defense is your immune response. This is very specific. As I mentioned, we will go into that lecture later on. You are not being tested on that in your second in-course exam. So if you look at this organism here, this pathogen, it needs to get through the first, second, and third lines of defense in order to cause an infection. The first line of defense being your intact skin or your mucous membranes. The second line of defense being things like inflammation or phagocytosis. And then your third line of defense, your most specific type of defense, is your immune system. Right? So it just categorizes those there again as either specific or non-specific. So your non-specific host defense mechanisms, they generally serve to protect your body against any harmful substances, that's innate or inborn resistance. There are other non-specific host defense mechanisms that include your mechanical and physical barriers, chemicals that are produced on the surface of your skin that are present in sweat, microbes that are present on the surface of your skin that may fight off invading particles. And then you have other things in your second line of defense like fever, inflammatory response, and phagocytes. So we have three major things that make up our first line of defense. First and foremost, all pathogens come into contact with our skin and mucous membranes, which act as the first physical barrier against any invading particle trying to enter into our bodies. Then we have cellular and chemical factors. Our skin can produce sweat. It can change temperature. We have cellular enzymes present on the surfaces of our skin. All of those act as chemical and cellular factors that help prevent organisms from causing infection. And then we can have microbial antagonism. This is where the indigenous microflora that live on our bodies, they prevent colonization of new arrivals because they do not want them to compete with them for their habitat or their site or their nutrients. So they produce lethal substances to stop the spread of those microbes. Then you have our second line of defense. Now, usually previous to this, when we talk about second line of defense, one of the only things that comes to mind would be your phagocytosis, right? However, you have many other things that assist in that second line of defense. We have the presence of many different types of proteins, example, transferrin. Transferrin binds to iron in your body, depriving the pathogens of that vital nutrient so they cannot survive. Fever, which is stimulated by pyrogenic substances. So generally, when our bodies are trying to fight off pathogens, you tend to have a fever. What this does is it augments the host defenses by stimulating your leukocytes to reduce the amount of iron present in your blood again, so that the organisms do not have a source of iron to survive. You can have interferons, which are antiviral proteins produced when a virus is present in your body. They help to prevent viruses by multiplying, and there are 3 different types: alpha, beta, and gamma. They would do different things. They are not virus specific, but they are usually species specific. So they would target specific species of viruses. And sometimes they can cause non-specific flu-like symptoms. So sometimes we experience flu-like symptoms, which not necessarily is because we have a virus, but it could be because of the presence of interferons in our body fighting off a virus or preventing the virus from multiplying. You can also have your complement system. Your complement system is a very intricate type of system, a way in which your body fights off diseases. What happens is a group of about 30 different proteins that is found in your blood plasma. It's complementary to the immune system, and they interact with each other and they form a cascade effect by which they help to destroy pathogens. The process by which they do that, they end up tagging your pathogens so that when the pathogens are circulating in the bloodstream, your phagocytes quickly identify them as foreign and they fight them off. And that process by which they are tagged is called opsonization, because there are specific opsonins that are deposited on those pathogens that tag them as foreign bodies. So your phagocytes or your white blood cells can very easily identify them and destroy them. You can have acute phase proteins. These are plasma proteins that rapidly increase in response to infection. They help to trigger inflammation. Cytokines, these are chemical mediators that are produced by cells in your body that allow cells to communicate with each other. So they help to basically attract phagocytes where infection is present, and the phagocytes to basically come in and kill off those pathogens. Or you can have inflammation, one of the most common types, right? Your body responds to a local injury by allowing inflammation to take place. During inflammation, what happens is there's an increase in the diameter of your capillaries, causing a lot of blood flow to the site of infection. Capillaries increase in permeability, allowing plasma proteins to escape from your blood vessels and collect at the site of injury. This exit of your leukocytes or white blood cells allow them to accumulate at the site of the infection and therefore fight off any invading particles that may be trying to enter into that site. The primary purpose of your inflammatory response is to localize an infection so that those phagocytes are not allowed into your bloodstream. It prevents the spread of them. It also helps to neutralize any toxins that are produced at the site so they do not enter your bloodstream. And it helps also to repair damaged tissue. There are 4 major signs of inflammation, which include redness, heat, swelling, and pain. So when you see an inflamed area, it's because your body is responding to try to localize that infection and not let spread take place. All of the plasma that escapes in that site is causing that site to become swollen or edematous, right? And again, the purposes would be to localize, prevent spread, destroy pathogens, and assist in healing processes. So inflammation is an extremely important second line of defense response. So inflammation is triggered by an injury. It causes vasodilation, it causes an increase in blood flow at the site. The capillaries become increased in permeability so that they allow your leukocytes to leak out into that region. It causes chemotaxis, or the attraction of more leukocytes, and phagocytosis, where your phagocytes accumulate to fight off any incoming or invading pathogens and toxins. This accumulation of fluid at the site is known as inflammatory exudate, and usually when that exudate is fighting off invading pathogens, it tends to form pus. So wherever you have pus at a site of inflammation, it would have detected the fact that your immune system was fighting off pathogens that were trying to enter. Some cases in many inflammatory responses, there is no exudate or no invading microorganisms, so you do not have that pus production. Right? If you do have microorganisms present, and there are some microorganisms that also increase the amount of pus produced at the site, so that they can identify what type of infection depending on how much pus production you have at a site of infection. And then finally, most common type of second line of defense is phagocytosis. As I mentioned, some of the proteins that are released by your body assist in how phagocytosis takes place. So your phagocytic white blood cells, which are called phagocytes, they surround and engulf foreign material in a process called phagocytosis. There are three major categories of phagocytes, which are your monocytes, lymphocytes, and granulocytes. And there are three types of granulocytes: eosinophils, basophils, and neutrophils. The most common phagocytes that you would find in the human body are your macrophages and neutrophils. So you see a neutrophil on your right there. When we looked at the slides, we saw a lot of macrophages, it's a lot of neutrophils that were present in the body, again, because those are most commonly present for phagocytosis. So there are 4 steps that take place in phagocytosis. Chemotaxis, where phagocytes are attracted by proteins that I mentioned before. So chemotactic agents attract phagocytes to the site of infection. Phagocytes attach to the object, in this case, the pathogen or the toxin. They surround that substance or object by forming pseudopodia or tiny false feet, causing the object to be engulfed into the phagocyte, and they fuse those objects with lysosomes inside of them to allow for digestion by digestive enzymes being present, right? So we have opsonization, where they are attracted to the invading particle. So they attract them based on the fact that they have opsonins present. The phagocytes attach to them. The pseudopodia extend around them, fuse to engulf them. The bacterial cell is then infused inside of the phagocyte. It's now called a phagosome. That phagosome binds with lysosomes inside of the phagocyte, and it causes it to be destroyed because of the digestive enzymes present within them. So this is the digestion phase of phagocytosis. In this image here, you can actually see leukocytes of a rat, which is actually killing invading Giardia trophocytes. So you're seeing the process by which they actually engulf them and start destroying them. Again, a scanning electron micrograph showing you phagocytosis. So you're seeing there the Giardia harmful substance being engulfed by the leukocyte and then broken down. So, as with all microorganisms, they also develop ways in which they can hide from phagocytes. They can use capsules, which cause them to be camouflaged. There are some bacteria that can produce an exoenzyme called leukocidin that can kill pathogens. There are other bacteria that cannot be destroyed in the phagolysosome. And the mechanism by which each pathogen evades digestion by these enzymes can differ, and in some cases, it is not fully understood. In other cases, there are some disorders that humans can be affected, that affect how their inflammation takes place or how their phagocytes work. So for example, some people suffer from leukopenia, where they do not have many leukocytes circulating in their blood. There are some other people that may be affected by diseases that cause their leukocytes to be less motile, or it prevents them from carrying out chemotaxis. Right? Inability of leukocytes to migrate in response to these agents may be related to a defect in the production of an enzyme, a protein called actin. Or you can have other types of disorders and additional factors that affect it. Additionally, host defense mechanisms can be affected by their nutritional status, if they have increased iron levels, stress, age, if they have other diseases like HIV, AIDS, or genetic issues, cancer, and so on. Okay, so that brings us to the end of this topic. As I mentioned,